In this lecture, we'll finish up Chapter 28 by covering adaptations for nutrition, defense, and reproduction in the protists. By the end of the lecture, you should be able to list and describe 4 methods of protist nutrition, list and describe the major types of protist defensive adaptations, and briefly describe asexual reproduction and the zygotic, sporic, and gametic life cycles. When it comes to nutrition, protists aren't quite as diverse as the prokaryotic bacteria, but there are still different ways that protists can obtain nutrition. One is phagotrophy. In phagotrophy, cells ingest particles by phagocytosis. Remember that phagocytosis is a form of endocytosis, where the cell moves the cell membrane to engulf a particle and then ingest it. Osmotrophs are also feeding on organic particles, but these particles are quite a bit smaller. Osmotrophs may include protists that are working as decomposers, and they may also include protists that are parasites and feeding on the cells of their hosts. Another mode of nutrition is photoautotrophs, which are performing photosynthesis, like your algae. The mixotrophs do both. They do photosynthesis and some type of heterotrophic feeding, such as phagotrophy or osmotrophy. There are diverse adaptations that protists use to protect themselves. Protists may shoot sharp projectiles from their cells, or they may produce flashes of bioluminescence. Bioluminescence, at first sight, may not seem like a defensive mechanism. After all, aren't they just advertising their presence? Well, bioluminescence may work in a couple of ways. One, it could startle a predator. Bioluminescence may also act like a beacon, announcing to the predators of the protist predator that there's food over here. Come over here and eat. Toxins are another common adaptation. Some examples are the toxin produced by PSP, or paralytic shellfish poisoning, that are produced by blooms of dinoflagellates. Domoic acid is a toxin produced by some species of diatoms. If the protist has a cell wall, that cell wall may be reinforced with cellulose or silica or calcium carbonate, making the protist more difficult to eat or digest. Life cycles are where protists really get creative. For the most part, protists have life cycles that incorporate both asexual and sexual stages. Let's take a moment to think about what might be the advantages of asexual reproduction. You can pause if you want more time to think. Well, an advantage of asexual reproduction is it's rapid. You don't have to find mates, you don't have to make specialized cells, so you can have rapid reproduction to take advantage of food and good living conditions. So what might be the advantages of sexual reproduction? Well, one is genetic diversity. Also in protists, the sexual part of the life cycle may involve a cyst or a similar stage for dormancy during less optimum environmental conditions. There are 3 different life cycles that are found in the protist groups, plus a few variations on those cycles, but for this lecture, we'll just stick to the basic patterns. The first one we are discussing is the zygotic life cycle. Let's take a moment to look at some of the more subtle details in this diagram. For instance, this diagram clarifies which are the haploid life cycles, so this is haploid, and which are the diploid part of the life cycle. Remember that haploid means the cells have 1 copy of each chromosome. Diploid means the cell has 2 copies of each chromosome. To put this into context, humans are diploid. Our body cells have 2 copies of each chromosome. Human gametes, sperm and eggs, are haploid. Sperm and eggs have 1 copy of each chromosome. So if we look at this diagram, the purple part of this diagram is showing us that most of the life cycle is actually in the haploid stage. So the haploid stage, haploid, or we can abbreviate it as N, is dominant. In this life cycle, there's only one diploid cell, right here. A diploid, shorthanded as 2N, dormant zygote forms. So note that most of the optimal season for this particular life cycle will be spent in the haploid stage. The formation of gametes that will then fuse to form a zygote may only be triggered near the end of the optimal season when environmental conditions become stressful. So let's just run over this one more time, make sure we've got it. If we start with our cell, here's our cell, Chlamydomonas, little cutie, haploid, reproducing by mitosis to increase the population. Then environmental conditions get bad. Maybe it gets colder or darker, or nutrient levels drop. And that triggers these haploid cells to start producing gametes. And the gametes may have a plus or minus mating stage. Gametes combine, so this is fertilization, to form our zygote. So in zygotic life cycles, oftentimes this zygote also forms that cyst stage that can hang out in the sediments or the water column until conditions get better. Then the zygote undergoes meiosis to produce our haploid cells again, and the life cycle repeats itself. Okay. In addition to being a marine biologist, I'm also a botanist. The sporic life cycle, also known as alternation of generations, is a life cycle that is characteristic of many seaweeds and of land plants. So in this example, we see one of the kelps. The key to understanding the sporic life cycle is that you actually have an alternation between 2 multicellular life stages. You have a multicellular sporophyte. So when I see the word sporophyte, I think multicellular plant. And I've also got a multicellular gametophyte. And when I hear gametophyte, I'm again thinking plant. So the sporophyte, let's look at our key. The sporophyte is diploid. Let's use our shorthand, diploid. So this multicellular seaweed here is a diploid organism, and in regions of the body, for kelps it's usually little patches on the blades, specialized cells will undergo meiosis to form spores. We don't call them gametes right now, they're spores. And the reason we don't call them gametes is because spores actually will grow into the gametophyte. Spores don't combine, they don't fertilize. So the spores will grow into the male gametophyte and the female gametophyte. So these are haploid multicellular organisms, and they grow. And eventually the female gametophyte produces an egg, and the male gametophyte produces sperm, and the sperm swim over and fertilize the egg, and now we have our zygote. And this zygote here is going to grow into our multicellular diploid sporophyte. So we have 2 multicellular stages, the sporophyte and the gametophyte, and 2 haploid cell stages, spores that grow into the gametophyte, gametes that combine to form the zygote, and then grow into the sporophyte. Then our last life cycle is the gametic life cycle. In the gametic life cycle, the haploid gamete stage is short-lived. Most of the life cycle is spent in the diploid stage. Diatoms are a good example of this life cycle. So diatoms are diploid. Let's get our diatom here. So diatoms are diploid, and they will divide by mitosis. So here's mitosis. They will divide by mitosis to make more diploid diatoms, and happily take advantage of good weather, good temperatures, and high nutrient levels. However, each time they divide, one of the diatom daughter cells is a little smaller. This is because the diatom cell wall is like a box with a lid on top of it. One side, when the box and the lid separate and form the sides of the new diatoms, one side is always smaller, and when diatoms are formed from that smaller side, the size of the diatoms continually gets smaller and smaller. So when diatoms reach a critical small size, or when environmental conditions get stressful, the cell undergoes meiosis to produce haploid gametes. So let's follow that where we are. Here we've got our diatoms. They're reproducing by mitosis to make more diploid diatoms. But then when they reach a small size or environmental conditions get bad, the diatom cell undergoes meiosis to produce my haploid gametes. Haploid gametes combine. So two gametes combine to form my diploid zygote. So the haploid stage is very short-lived. This diploid zygote then can either undergo mitosis to form diatoms again, or it can form a cyst and wait until better conditions if it's the end of a growing season. Note also that that zygote is larger than the diatoms that form the gametes. That means when it undergoes mitosis, it's going to restore the original size of the diatoms. Life cycles are tough. I usually call those my Advil lecture. A little practice, review, and I suggest looking up animations or other resources online to kind of constantly reinforce those concepts. So our big ideas for this lecture: protists have diverse adaptations for nutrition, including phagotrophy, osmotrophy, photosynthesis, and mixotrophy. They protect themselves by sharp projectiles, light flashes, toxic compounds, and tough cell coverings. And their life cycles are diverse and frequently include both asexual and sexual reproduction. So thank you for joining me, and I'll see you in our next lecture, which will be our case study.